Welcome, now I presenting a video about the hurdle technology. What is the hurdle? Each hurdle aims to eliminate, inactivate or at least inhibit unwanted microorganisms. Common salt or organic acids can be used as hurdles to control microbials in food. Many natural antimicrobials such as nicene, natamycin and other bacteriocins and essential oils derived from rosemary or thyme also work well. The majority of foods rely on the application of a combination of preservative elements known as hurdles to maintain their microbiological safety and stability as well as their sensory and nutritional quality. The main factors are temperature, high or low, acidity, pH, water activity, AU, redox potential, A, and preservatives, sorbate, nitrite, and sulfite. What is hurdle technology? Hurdle technology is a process by which every preservation parameter is used at an optimum level in order to achieve maximum microbial inactivation by the amalgamation of two or more such parameters, so that damage to sensory properties of food is kept at a minimum. Hurdle technology is a method of ensuring that pathogens in food products can be eliminated or controlled. Liestner said as an intelligent combination of hurdles which secures the microbial safety and stability as well as the organoleptic and nutritional quality and the economic viability of food products. Objectives of preservation: to ensure the safety of food from microbes, to prevent the spoilage of food, to enhance the keeping quality of food, to control foodborne infections and intoxications, to extend the shelf life of food, to reduce economic losses. Principle of hurdle technology. Conventional food preservation methods such as heating, chilling, freezing, freeze-drying, drying, curing, salting, sugar addition, acidification, fermentation, smoking or oxygen removal are used in making food safe and stable. Liestner, 1992. The microbial safety, stability and sensory attributes of many processed foods including traditionally processed depend on the combined effects of many hurdles. Liestner, 2000. The hurdle effect is the most fundamental importance for the preservation of foods, since the hurdles in a stable product control the microbial spoilage, food poisoning and desired fermentation processes. Aspects of hurdle technology. The hurdle technology affects the physiology and growth of microorganisms in food. There are mainly four major mechanisms by which hurdle technology affects the growth of microorganisms in foods. 1. Homeostasis. 2. Metabolic exhaustion. 3. Stress relaxation. 4. Multi-target preservation of food. 1. Homeostasis. It is the process that maintains the stability of the living cell's internal environment in response to the changes in external environment. Some of the examples of homeostasis in the body are regulation of temperature and balance between acidity and alkalinity, pH. These factors are prerequisite feature of living cells, and this applies to higher organisms as well as microorganisms. In case of food preservation techniques, if somehow the homeostasis of the pathogens are disturbed, then they will not be able to proliferate themselves. They will remain in the lag phase or even die until their internal body temperature is maintained or recovered. Disturbing the homeostasis of the microorganisms by various hurdles eventually results in the death of the spoilage-causing microbes, thereby protecting the food product from microbial spoilage. 2. Metabolic exhaustion. This aspect of hurdle technology deals with auto-sterilization of food. It has been observed that the spore counts in hurdle technology treated food products actually decreases during storage, especially at ambient temperature. The microbes in the hurdle treated stable products uses their energy for homeostasis, thereby becoming metabolically exhausted. This leads to auto-sterilization of food products. 3. Stress reaction. Some microbes acquire resistance or may become more virulent under stress conditions as they synthesize stress shock proteins. The synthesis of stress shock protein is affected by several factors like pH, water activity, ethanol, heat, etc. The different responses of microbes under stress conditions might hamper the food preservation. Exposure to multiple stresses simultaneously activates the energy-utilizing synthesis of several stress shock proteins, in turn making the microbes metabolically weak. Therefore, multi-target preservation of foods could be an efficient approach towards reducing the synthesis of stress shock proteins and in food preservation. 4. Multi-target preservation of food. It is a very important aspect for efficient and effective preservation of targeted food material. Hurdles applied in the targeted food material might not just have effects on microbial stability, but they could act synergistically. Synergistic effect could be achieved in the targeted food if the hurdles affects different targets such as pH, AOA, enzyme systems simultaneously within the microbial cell and thus disturb the homeostasis of the microbes, rendering it difficult for the microbes to synthesize different stress shock proteins and to maintain their homeostasis. Therefore, the application of several hurdles simultaneously would lead to an optimal microbial stability and effective food preservation. Types of hurdle technology. There are mainly 3 types. 1. Physical. 2. Physiochemical. 3. Microbial. 1. Physical. Ionizing radiation, low temperature, chilling, freezing, aseptic packaging, high temperature, blanching, frying, baking, extrusion, evaporation, pasteurization, sterilization, modified atmosphere, electromagnetic energy, radio frequency, high electric fields, pulse magnetic fields, microwave, ultrasonification, packaging films, ultra-high pressures, photodynamic inactivation, ultraviolet radiation. 2. Physiochemical. There are some chemicals such as sodium nitrate, sodium sulfite, sodium nitrite, potassium sulfite, phosphates, salt, spices and herbs, carbon dioxide, ozone, oxygen, organic acids, surface treatment agents, phenols, ethanol, lactic acid, low redox potential, low pH, low water activity, lactoperoxide, smoking, Maillard reaction products. 3. Microbial. Bacteriocins, protective cultures, competitive flora, antibiotics. Application in dairy products. Hurdle technology has been applied in many dairy products to enhance their shelf life. Example, paneer is a highly perishable product. It was reported that the freshness of paneer remains intact only for 3 days at refrigeration temperature. Bhattacharya et al., 1971. At room temperature, paneer does not keep good for more than 1 day. In order to increase the shelf life of paneer, additives, modification in paneer manufacturing process, surface treatments and packaging materials have been recommended by various workers. Several hurdle techniques have been used by researchers for increasing shelf life of paneer. One of the hurdle technology involved mild heat treatment, minor reduction in water activity 0.95 and acidification pH 5.0 to extend their shelf life of paneer to 14 days at 30 degrees Celsius. Use of 1% each of sodium chloride, sucrose and glycerol to decrease the water activity of paneer led to shelf life extension of paneer. Rao and Patil, 1992. Fish and fish products. While applying hurdle technology for particular food product, the selected processing method, preservation and packaging will affect the spoilage mechanism and leads to the major quality deterioration fish and fishery product. For example, the fish preserved by cold smoking process. The combination of hurdles includes salt addition, mild thermal treatment and storage at low temperatures, In this case, the quality deterioration is mainly attributed to microbial spoilage, resulting in sensory modifications and thus organoleptic rejection. However, when same food products stored at sub-zero temperatures, then the quality deterioration is correlated with physical and chemical reactions, viz., dehydration and lipid oxidation. These are major factors that defining duration of shelf life of the final products. Fruit-derived product. Several conventional hurdle strategies are effectively used along with the novel ones for the preservation of various fruit products. Some of the hurdles applied in fruit processing includes UV light, pulsed light, PL, ultrasound, US, and high hydrostatic pressure, HHP. Hurdle or combined technology is also applied in the preservation of high-moisture fruit products such as peach, pineapple, papaya, mango and banana. The technology is based on the combination of heat treatment and addition of antimicrobials. That is the hurdle technology. Please correct me if I am wrong. Thank for watching and subscribe the channel. Thank you.